Hyperreal Vault, Elite Biosystems Edition, by Gerard King, gerardking.dev.d, hyperrealistic imaging of biological systems, including high-resolution simulations of cellular interactions, genetic mutations, and intricate biological processes. Research and analysis. Category. Here are the primary categories for Hyperreal Vault. Elite Biosystems Edition. Cellular and molecular biology, cell structure and function, cellular communication, genetic expressions, molecular interactions, signal transduction pathways, immunology, immune response dynamics, T-cell and B-cell interactions, antigen presentation, cytokine release, pathogen recognition and elimination, genetics and genomics, DNA replication and repair, gene regulation and expression, epigenetic modifications, genetic mutations and variations, CRISPR and genetic engineering, neuroscience, neuronal structure and function, synaptic transmission, neural networks and pathways, neurotransmitter dynamics, brain tissue microarchitecture, microbiology, bacterial cell structure, viral particles and infections, microbial pathogenesis, antibiotic resistance mechanisms, host microbe interactions, pathology, disease mechanisms at cellular level, tumor microenvironment, inflammatory responses, cellular responses to injury, pathogen-induced cellular damage, pharmacology, drug-cell interactions, mechanisms of drug action, drug metabolism and detoxification, receptor binding and activation, pharmacokinetics and pharmacodynamics, developmental biology, embryonic development stages, stem cell differentiation, tissue morphogenesis, developmental gene regulation, cellular migration and patterning, biochemistry, enzyme structure and function, metabolic pathways, protein folding and dynamics, lipid membranes and transport, biomolecular complexes, advanced imaging techniques, electron microscopy innovations, high-fidelity molecular rendering, 3D imaging of cellular structures, advanced fluorescence imaging, quantitative imaging analysis, cellular communication, image title, synaptic transmission excellence. Description. This high-resolution image captures the precise moment of synaptic transmission between two neurons, highlighting neurotransmitter release and receptor binding. Macro details. Synapse. Clearly defined synaptic cleft and vesicles. Axon terminal, detailed with synaptic vesicles. Micro details. Neurotransmitters, visible release of neurotransmitters, receptors, detailed postsynaptic receptors. Background. Neural networks and glial cells. Production metadata. Imaging technique, advanced electron microscopy and synaptic rendering. Resolution, 8K rendering, high-fidelity synaptic level. Hashtags, hashtag synaptic transmission, hashtag neural communication, hashtag neuroscience imaging. Genetic expressions. Image title, transcription and translation dynamics. Description. This image shows the complex process of transcription and translation within a eukaryotic cell, detailing RNA synthesis and protein assembly. Macro details. Nucleus. Detailed RNA transcription at chromatin sites, cytoplasm, visible ribosomes translating mRNA. Micro details. RNA polymerase. Detailed RNA polymerase activity. Ribosomes. Ribosomes actively translating mRNA. Background. Cytoplasmic matrix with various cellular elements. Production metadata. Imaging technique. Electron microscopy with molecular rendering. Resolution. 8K rendering. High-fidelity genetic level. Hashtags. Hashtag transcription, hashtag translation, hashtag genetic expression. Molecular interactions. Image title. Protein-protein interaction precision. Description. This image captures the interaction between two proteins at the molecular level, highlighting binding sites and conformational changes. Macro details. Protein complexes. Clearly defined protein structures. Binding sites. Detailed binding interactions. Micro details. Amino acids. Visible side chain interactions. Conformation. Detailed conformational changes. Background. Cellular environment with other interacting molecules. Production metadata. Imaging technique. Molecular rendering and electron microscopy. Resolution. 8K. Rendering. High-fidelity molecular level. Hashtags. Hashtag protein interaction. Hashtag molecular binding. Hashtag biochemistry imaging. Signal transduction pathways. Image title, Intracellular Signaling Cascade. Description. This image illustrates a signal transduction pathway within a cell, showcasing receptor activation, second messenger release, and downstream effects. Macro details. Receptors. Activated receptors on the cell membrane. Pathways. Clearly defined signaling molecules and pathways. Micro details. Second messengers. Detailed view of second messenger molecules. Kinases, active kinases and phosphorylation events. Background. Cytoplasmic matrix with detailed cellular structures. Production metadata. Imaging technique. Advanced electron microscopy with molecular rendering. Resolution, 8K rendering. High-fidelity signaling level. Hashtags, hashtag signal transduction, hashtag cell signaling, hashtag intracellular pathways. High-fidelity molecular rendering. Image title, Precision in Molecular Rendering. Description. This high-resolution, hyper-realistic 3D image illustrates high-fidelity molecular rendering. The image showcases complex protein structures and interactions at the molecular level with exceptional detail and accuracy. Macro details. Protein structures. Detailed complex protein structures. Molecular interactions. Highlighted binding sites and conformational changes. Micro details. Atomic details. Intricate atomic details of molecular interactions. Background, gradient to enhance the visibility of molecular structures. Production metadata. Imaging technique, high-fidelity molecular rendering. Resolution, 8K rendering, high-accuracy molecular level. Hashtags, hashtag molecular rendering, hashtag protein structures, hashtag advanced imaging. 3D imaging of cellular structures. Image title, Comprehensive Cellular Architecture. Description. This high-resolution, hyper-realistic 3D image showcases various cell types using advanced 3D imaging techniques. The image highlights the unique shapes, organelles, and interactions within a tissue matrix, providing a comprehensive view of cellular architecture. Macro details, cell types, exquisite detail of various cell types, organelles, highlighted organelles and their interactions. Micro details, cell shapes, unique shapes and spatial relationships of cells. Background, subtle gradient to enhance visibility of cellular structures. Production metadata. Imaging technique, advanced 3D imaging of cellular structures. Resolution, 8K, rendering, high-fidelity cellular level. Hashtags, 3D imaging, hashtag cellular structures, hashtag advanced imaging. Advanced fluorescence imaging, image title, illuminated cellular interactions. Description. This high-resolution, hyper-realistic 3D image uses advanced fluorescence imaging techniques to highlight various cellular components with fluorescent markers. The image showcases the intricate details of cellular structures and interactions. Macro details. Cellular components. Various cellular structures highlighted with fluorescent markers. Organelles. Specific proteins and pathways illuminated. Micro details. Fluorescent markers. Exceptional contrast and clarity of fluorescent signals. Background, dark gradient to enhance the visibility of fluorescent signals. Metadata. Imaging technique, advanced fluorescence imaging. Resolution, 8K rendering, high-fidelity fluorescence level. Hashtags, hashtag fluorescence imaging, hashtag cellular fluorescence, hashtag advanced imaging. Quantitative imaging analysis. Image title, Precision in Quantitative Imaging. Description. This high-resolution, hyper-realistic 3D image illustrates quantitative imaging analysis of cellular structures. The image showcases detailed measurements of cellular components, highlighting quantitative data overlay on cellular images. Macro details. Cellular components, detailed measurements of cell size, organelle distribution, and molecular concentrations. Micro details. Quantitative data. Overlay of precise measurements and data on cellular images. Background. Clean gradient to emphasize the quantitative data. Production metadata. Imaging technique. Quantitative imaging analysis. Resolution, 8K rendering, high-fidelity quantitative level. Hashtags, hashtag quantitative imaging, hashtag cellular analysis, hashtag advanced imaging.